Hello everyone, welcome to Ritika's Online Teaching Academy. Today I will talk about a new lecture series on DNA structure and DNA packaging. So this is the first lecture on this topic and it is also a chapter 6 of NCERT book, which is molecular basis of inheritance. So let me share my screen with you. Okay, now let's talk about DNA structure and DNA packaging. So first, what is DNA? We all know that DNA is a genetic material and the full form of DNA is deoxyribonucleic acid. So DNA structure is generally a long polymer of deoxyribonucleotides and it encodes the instructions for the formation of other cellular components in a living system. And also it provides a template for the production of identical DNA copies to be distributed to the progeny when the cell divides and thus maintains individual species. So for an individual species, let's say mammalian species, so the DNA which is inherited from their parents to the progeny maintains the qualities or the properties of their parents in the progeny as well. So this is the feature of the DNA which is used to maintain individual species. Now, what is the length of DNA? Length of DNA is generally described as number of nucleotides or base pairs present in it. So, what are nucleotides? Nucleotides are the basic unit in the structure of the DNA. We'll talk about it just in few slides. So, haploid content of human DNA contains around 3.3 into 10 raised to 9 base pairs. So, we can see the number of base pairs in human DNA is around this much. In E. coli, DNA is a single molecule and contains about 4.6 into 10 raised to 6 base pairs, or we can say 4.6 million base pairs. So here you can see that the number of base pairs as the complexity is decreasing between the organisms like human is more complex compared to the E. coli, which is a simple bacteria. The number of base pairs gets decreased. In Bacteriophage lambda, it is around 48,502 base pairs, while in Bacteriophage 5174, it contains only around 5,386 nucleotides. So this is about few examples about the length of DNA. Now we will deal with the structure of a polynucleotide chain. As in the first slide, I have told you that DNA length or DNA consists of a chain of nucleotides. Means there are so many nucleotides. If we say there are two nucleotides, then it will be called as a dinucleotide. When there are many nucleotides, then we can say it as a polynucleotide. So what is a nucleotide structure? That nucleotide structure generally contains a nitrogenous base, a pentose sugar, and a phosphate group. So these are the three basic units in the formation of a nucleotide. Now, what are nitrogenous bases? Nitrogenous bases which are found in DNA are generally of two types, purines and pyrimidines. Purines examples, what purines are found in DNA? They are adenine and guanine. Pyrimidines are cytosine, uracil, and thymine. So cytosine is common to both DNA and RNA. Means in both DNA and RNA, cytosine is found, while thymine is found in DNA, and the another chemical name of thymine is 5-methyl-uracil also. So uracil is found in place of thymine in RNA. So in RNA, in place of thymine, uracil nitrogenous base is found. What type of pentose sugar? Pentose sugar is also different in DNA and RNA. In the case of DNA, it is called as a deoxyribose, while in the case of RNA, it is a ribose sugar. So this is a polynucleotide or single polynucleotide chain of a DNA. Here the chemical structures are shown. This pentose ring or pentose sugar, it is a pentose sugar. It is a deoxyribose and not ribose. Why I am saying it as a deoxyribose? Because at its 2' position, this is 1', this is 2', 3', 4', and 5' position of carbon in this pentose ring. So at the 2' position, hydrogen is found. So it is called as a deoxyribose in case of DNA. And when in place of this H, hydroxyl group or OH group is present, then it is called as a ribose sugar and is present in RNA. So at the 1' position of this pentose sugar, this base, nitrogenous base is present. C stands for cytosine, A stands for adenine, and G stands for guanine. At the 3' position here you can see this phosphate group is present and it is called as a diester. Why? Because it has a composition here you can see at the 3' carbon is present. So POOC. So it is one ester and here you can see POOC at the connecting at the 5' carbon to the of the next pentose sugar. So it is forming a diester link between one pentose to the, or you can say between the one nucleotide and the other nucleotide. Now why we are saying it as a nucleotide? Because at the 5' position of the carbon, phosphate group is present. When there is no phosphate, and there is only sugar and the nitrogenous base, then it is a nucleoside. And when at the 5' position a phosphate group is present, then it is a nucleotide. So these nucleotides are linked by the phosphodiester bonds. Here in this polynucleotide chain, I have shown 3 nucleotides. In first nucleotide, cytosine is present at the 1' position and is linked by the N-glycosidic bond. While in the second nucleotide at the 1' position, adenine is present and is linked by the N-glycosidic bond. So bases are linked to the pentose sugar at their 1' position by means of N-glycosidic bonds. So one more point is there that at the 5' position, the phosphate group is present. While at the 3' end, carbon is attached, carbon of the pentose ring is attached to the hydroxyl group. So 3' end of this polynucleotide chain is formed by the hydroxyl group, while the 5' end is formed by the phosphate group. So you can see that these two ends are containing different functional groups, and so they are different. So we can say that the 5' position phosphate free group is present, while at the 3' position hydroxyl free group is present. So this is a one nucleotide or polynucleotide chain of the DNA. Now next, so we can now summarize what is a nucleoside: a molecule with a phosphate group, without a phosphate group, and it consists of a nitrogenous base and a pentose sugar only. While in a nucleoside structure, a nitrogenous base is linked to a pentose sugar through an N-glycosidic linkage. Then these nucleosides are named as adenosine, guanosine, uridine, and cytidine when they are present in RNA, while they are named as deoxyadenosine, deoxyguanosine, deoxythymidine, and deoxycytidine when they are present in DNA. I have told you about the difference between the adenosine and guanosine and the guanosine only. So in the case of RNA, there is no deoxy, while in the case of DNA, deoxy we have to add due to the difference in the group which is attached at the 2' position of the pentose sugar. Now here the nucleotide and nucleoside structure is shown. In the first structure, which is a structure of deoxyadenosine. Why we are saying it as deoxyadenosine? Because at the 5' phosphate group is attached. This pink color portion which is excluding the phosphate group, is called as a nucleoside, because at the 1' position adenine is attached, hence it is called as deoxyadenylate or deoxyadenosine 5' monophosphate, and the symbol which are used for this is, we can simply write it as deoxyadenosine or deoxyadenosine monophosphate, and in the case of that RNA we can write it as an adenine. So this is deoxyadenosine nucleoside, while the nucleotide is deoxyadenylate. Okay. So without a phosphate group, it is a nucleoside, and with a phosphate group, it is a nucleotide. So the second structure is D of deoxyguanylate or deoxyguanosine 5' monophosphate. We can simply write it as DG or DGMP, and we can, in a short form, we can say it as a deoxyguanosine. The third structure is for deoxythymidine, while the fourth structure for deoxycytidine. So what we have learned till now, that you can see in all the structures, the bases are attached to the 1' position of the pentose sugar through the N-glycosidic bond. At the 5' position of the pentose sugar, phosphate group is there in order to form a nucleotide. And the other thing which you can see, at the 2' position of the pentose sugar, hydrogen is present. Hence these are the nucleotides of the DNA. And when in place of H hydroxy group is present, then they are the nucleotides of RNA. Now here two polynucleotide chains, or you can say the double helix structure of the DNA is shown. See, one strand is 3' to 5' polarity, while the other strand is having 5' to 3' polarity. And the groups which attaches to these ends are also different. Okay, so here you can see at the 5' position, no OH should be attached, while here at the 3' position, phosphate. Here OH and here phosphate. So both the ends are containing the different functional groups, and the polarity of the two strands are opposite to each other. One is 5' to 3', while the other one is 3' to 5'. So, and the sugar, these nucleotides are attached by the phosphodiester linkage, all these nucleotides. Now bases, nitrogenous bases are projecting towards the inside, while the sugar and phosphate are on the are forming the backbone of the polynucleotide chain, or you can say are forming the backbone of the DNA double helix. So these bases, here you can see thymine is attached to the adenine through the two double bonds, and these two bonds are actually hydrogen bonds. So hydrogen bonds is shown with the help of this dotted structure. We know that when hydrogen bond will form, the hydrogen bond would be formed between more electronegative atom and the hydrogen atom. So in this NH2 group, this hydrogen is participating with the oxygen with the help of one hydrogen bond. Here you can see hydrogen is attached with the more electronegative nitrogen with the hydrogen bond. So these two are electronegative, more electronegative compared to that of hydrogen, hence forming the two hydrogen bonds. Why in the case of, you can see, guanine and cytosine, there is the presence of three hydrogen bonds. The same concept of hydrogen bond, more electronegative oxygen is forming bond with hydrogen of the cytosine, and here H is forming hydrogen bond with nitrogen, and H is forming hydrogen bond with oxygen. So in the same way, you can see between the guanosine and cytosine, three hydrogen bonds are there, while between the adenine and thymine, two hydrogen bonds are there. So the questions, I have taken some questions from the NEET exam which are generally asked. Purines found in both in DNA and RNA are, the first option is adenine and thymine, the second option is adenine and guanine, third is guanine and cytosine, while the fourth one is the cytosine and thymine. So as we have studied, the purines are adenine and guanine. So this option is correct. Now the next question which was asked is bases in DNA structure are held by hydrogen bonds. So the type of these bonds are covalent bonds, non-covalent bonds, van der Waals forces, or the ionic bonds. So these hydrogen bonds are not covalent bonds, neither they are ionic bonds nor they are van der Waals forces. So these are what in the category of non-covalent bonds. So now we will study the salient features of the double helix structure of DNA. So the structure which I have shown you earlier and all those are based on some experiments or observations and the propositions given by different scientists. So in 1953, James Watson and Francis Crick proposed the double helical or double helix model of DNA. What was that? As we have seen, there are two polynucleotide chains with the sugar phosphate backbone and the bases projects inside. We have seen there in the earlier structure that sugar phosphate backbones are phosphates are forming the backbone while the bases are present in the inside. So anti-parallel polarity, one chain has 5' to 3' polarity while the other chain is having 3' to 5' polarity. And we have also seen that the bases on the two strands are joined or paired by hydrogen bonds. Between adenine and thymine, there are two hydrogen bonds, while between guanine and cytosine, there are three hydrogen bonds. So this also generates a uniform distances between the bases in the two strands. So this model is also based on the observations made by Erwin Chargaffs. What was his observation? That the ratios between the adenine and thymine and guanine and cytosine is constant, and that equals to 1. It means that adenine would be equal to thymine, while that of guanine content is equal to the content of cytosine. Okay. Now the other observation or assumption which was made was the two chains of DNA are coiled in the right-handed fashion. Means their coiling is towards the right hand. And there are around, they have estimated the number of base pairs as well as the distance between the base pairs also. So that there are around 10 base pairs in each turn. And the pitch of the helix is 3.4 nanometer. What do we mean by the pitch of the helix? Pitch of the helix stands for the rise or height of one helical loop. That I will show you in the next slide with the help of a diagram. The distance between a base pair is approximately 0.34 nanometer. Now, here we come to the point of the stability. How the helix or DNA helix is stable inside a nucleus. So the stability of helix is due to the presence of thousands of hydrogen bonds between those bases, as well as between the phosphate groups and other water molecules present. So that will maintain the stability of the helix, but this is the one factor. The other stability is given by the stacking of the planes of base pairs over the other in the helix, which is due to the presence of hydrophobic and van der Waals forces between their planes. Okay, so there is non-covalent interactions between the planes of the base pairs and the hydrogen bonds presence in the helix, which lead to the stability of the DNA. Now, in this picture, you can see that between the two base pairs, there is a distance of about 0.34 nanometer, while the pitch of the helix, or you can say the height of one loop or rise of one loop is around 3.4 nanometer or 34 angstrom. And the number of base pairs in one turn or helical loop is around 10 base pairs. Now we'll deal with the packaging of DNA helix. DNA is not a very small molecule. It is having a big length, and how that length of DNA or such big mobile molecule is packaged, or you can say is constricted to a very small volume of nucleus. We'll see that. So that comes under the packaging of DNA helix. So first point is length of DNA double helix in a typical mammalian cell. We can calculate it by the total number of base pairs multiplied by the distance between the two consecutive bases. The total number of base pairs in the mammalian cell is around 6.6 into 10 raised to 9 base pairs, and the distance between the two consecutive bases is around 0.34 into 10 raised to minus 9 meter per base pair. So that comes around 2.2 meters. Now you can see how much big its length is. So how it is constrained within a small nucleus volume which is around only 10 raised to minus 6 meter. The answer is its complex packaging. So DNA is packaged inside a nucleus and in eukaryotes, and this packaging is complex. So there are some proteins which assist or help in packaging of the DNA inside the nucleus. So what are these proteins? These are generally positively charged basic proteins. And why we are saying it as positively charged basic proteins? Because of the abundance of positively charged amino acids which are constituting the protein structure. And these positively charged amino acids are generally lysine and arginine. They are found in abundance, hence they are positively charged. And such type of proteins are called as histones. Okay, now in this structure, you can see these beads, yellow colored beads. These are generally nucleosomes, and the blue color rope-like structure which is wrapped around this nucleosome or bead is DNA, negatively charged DNA. So the wrapped portion of the negatively charged DNA is around 146 base pairs, while the remaining means total DNA which is wrapped and forming the linking region of DNA is around 200 base pairs. In that, 146 base pair is wrapped around the nucleosome, while the remaining four base pairs around um, around 146 is wrapped around this nucleosome, while the remaining is forming the linker DNA of the nucleosome. Okay, so this bead or the nucleosome is generally formed by the histone core, and these histone core are positively charged basic proteins. And this linker DNA of nucleosome also consists of one histone particle. So how many types of histones are there? And there are generally 5 different types: H1, H2A, H2B, H3 and H4. So we will discuss it. So this H1 histone is found in the linker region. This loop you can see here, it is found. In this linking region, this H1 histone is found. While H2A, H2B, H3 and H4 is found in this core region or this octamer region, core of the histone molecule. Now here you can see how the chromosomal DNA, the DNA in the chromosome is packaged. So a small portion of chromosome, when you enlarge, you can see the condensation of chromatin or the DNA in the chromatin. Now a small portion of this chromatin, if it is enlarged, you can see that the earlier structure, the DNA is wrapped around the nucleosome. So this beaded string is generally a part of this small portion. When the small portion of this nucleosome coiled in a chromatin fiber is enlarged, you can see the wrapped DNA around the histone. And when a small portion of this wrapped DNA is enlarged, you can see this double helical structure. So this very large double helical structure, how it is compacted in a small nucleus with the help of these histone proteins, we have seen. So there is a higher level of compaction in eukaryotes. So histone form a unit of 8 molecules, means histone octamers are there. How this octamer is formed? 2 copies each of H2A, H2B, H3 and H4 are there. So total 8 molecules of histones are there. Now the negatively charged DNA wraps around the histone octamer to form nucleosome. A nucleosome contains around 200 base pairs of the helix. Okay, we have seen that total DNA helix which is around the nucleosome or which form the nucleosome structure is around 200 base pairs, out of which 146 is wrap. Okay. Okay. Now next is nucleosome forms the repeating unit of the chromatin in the nucleus, and chromatin is generally what? It is a thread-like stained structure in the nucleus. So by the staining methods with the agents such as fusion agent, the chromatin structure can be seen in the nucleus and which looks like a thread-like stained structure. So in that stained structure, some beaded appearance would be seen, and those beads are generally nucleosome, which can be observed under the electron microscope. In prokaryotes such as E. coli, DNA is held with the help of some positively charged proteins in a region called as nucleoid. So in E. coli also, where around 4.6 million base pairs are there, they are also compacted. And how they are compacted? They are also, their compaction is also done by some kind of positively charged proteins, and they are constrained in a region called as a nucleoid because there is no such well-defined nucleus in E. coli. So these nucleosomes beads are packaged into the chromatin fibers as we have seen in the previous diagram. Okay, so in the chromatin fiber they are compacted. Okay, next, at the metaphase stage of cell division, chromatin fibers gets more coiled and condensed. So we know that the cell cycle is having different stages. So at the metaphase stages, these chromatin fibers further undergo coiling and condensation. So for more coiling, there are some other types of proteins apart from histones. They are called as non-histone chromosomal proteins, NSC are there. Okay. So when they are stained with some staining reagent such as fusion reagent, there are two differentiated portions there. So what are those? Euchromatin and the heterochromatin. Euchromatin will stain lightly in color, and they are also called as transcriptionally active, while the heterochromatin will appear as a darkly stained portion of the chromatin and it is transcriptionally inactive. So in NEET 2017, the question based on these histone protein was asked, and which is as follows. The association of histone H1 with a nucleosome indicates what? Transcription is occurring, DNA replication is occurring, the DNA is condensed into chromatin fiber, or the DNA double helix is exposed. So it will not show the transcription is occurring, neither it will show the DNA replication because histone H1 is used in the compaction and is a part of the linker DNA. So the option C, the DNA is condensed into the chromatic fiber, is correct. Okay, and the DNA double helix is exposed is also a wrong one answer. Okay. So now we have seen, we have learned about the structure of DNA and how it is packaged inside the nucleus. In the next lecture, I will talk more on the DNA, its history, and how it was discovered and all. So do like, share and subscribe my channel in order to get more lectures in biology, and that would be helpful to you for clearing NEET 2021-22 exam. So all the best. Thank you.